Hello, I am Dr. Nasmani from ASPJ Mat, and this is the first session of Physiology, concept bridge building. The concept bridge idea is to give you brief details about the basics of physiology. Similar sessions for concept bridge in other subjects are also being telecast in the same channel. Please visit the channel youtube.com/acpgmed. If you got account, please make friends with facebook.com/acpgmed. That is the channel address. Please note down. Let us start with the physiology concept bridge. Cell physiology. Plasma membrane. The structure of the plasma membrane allows the separation and creation of distinct molecular environments within cells. The lipid bilayer is similar to thin layers of oil surrounding fluid ozone. Thus, the lipid bilayer divides the cell into functional compartments. The fluid mosaic model is the accepted view of the molecular nature of the plasma membranes. The model proposes that proteins traverse the liquid bilayer and are incorporated within the lipids. Proteins and lipids can move freely in the plane of the membrane, producing the fluid nature of the membrane. Plasma membrane is composed of phospholipids and proteins. The membrane lipids can be classified into three major classes: phospholipids, Sphingolipids, and cholesterol. Membrane lipids are phospholipids, Sphingolipids, and cholesterol. The phospholipids are the most abundant membrane lipids. They have a bipolar nature or amphipathic nature, contain charged head group and two hydrophobic tails, which are water-insoluble and non-charged. Charged head group and two hydrophobic tails. The hydrophobic tail face each other, forming a bilayer and exposing the polar head group to the aqueous environment on either side of the membrane. Sphingolipids have an amphipathic structure similar to phospholipids that allows them to insert into membranes. These lipids can be modified by the addition of carbohydrate units at their polar end, creating glycosphingolipids in the brain cells. Cholesterol is the predominant sterol. There is unsaturated alcohol found in animal and plant tissues in human cells. Cholesterol is the predominant sterol in human cells. It increases the fluidity of the membrane by inserting itself between phospholipids, improving membrane stability. Clinical correlation is Tay-Sachs disease. The accumulation of glycosphingolipid associated with Tay-Sachs disease causes paralysis and impairment of the mental function. Membrane proteins that span the liquid bilayer are called the integral membrane proteins, whereas those associated with either the inner or the outer surface of the plasma membrane, are known as peripheral or lipid anchored membrane protein. The inner is the peripheral and outer is the lipid anchored membrane protein. Majority of the integral membrane proteins span the bilayer through the formation of helices, a group of 20-25 amino acids twisted to expose the hydrophobic portion of the amino acids to the lipid environment in the membrane. Please see the figure. Protein content of the membrane varies from less than 20% for myelin, a substance that helps the propagation of action potential, to more than 60% of the liver cells which perform metabolic activities. Protein is 20% in myelin, 60% in liver cells. So that is the picture of the membrane protein showing the plasma peripheral membrane protein, the cholesterol molecules, the integral membrane proteins, and the two layers. Cellular proteins act as receptor sites for antibodies as well as hormone, neurotransmitter, and drug binding site. Enzymes bound to the cell membrane are often involved in phosphorylation of metabolic intermediates. Carrier proteins in the membrane transport materials across the cell membrane. Carrier proteins in the membrane transport materials across the cell membrane. Membrane channels allow polar charged ions, sodium, potassium chloride, and calcium to flow across the plasma membrane. Ion channel gates regulate ion passage and are controlled by voltage, that is called voltage gated, and ligands, which are called ligand gated, or mechanical means, that is called mechanically gated. So the ion channel gates are voltage gated, ligand gated, or mechanically gated, and through them the polar charged ion, sodium, potassium chloride, and calcium flow. Plasma membranes act as selective barrier to maintain the composition of the intracellular environment. Passive transport or diffusion involves transport of solutes across the plasma membrane due to the substance concentration gradient. The term passive implies that no energy is expended directly to mediate the transport process. Passive transport is simple diffusion of substance that can readily penetrate the plasma membrane, as is the case for oxygen or carbon dioxide. Passive transport is the only transport mechanism that is not carrier mediated. Substances diffuse because of their inherent random molecular movement, that is following the principles of Brownian movement. Diffusion across membranes occurs if the membrane is permeable to the solute. The net rate of diffusion J is proportional to the membrane area A and solute concentration difference, that is C1 minus C2, and permeability P of the membrane. So diffusion is measured using the formula J is equal to Pa C1 minus C2. Facilitated diffusion is the transport of a substrate by a carrier protein down its concentration gradient. Facilitated diffusion is required for substrates that are not permeable to the lipid bilayer and is faster than simple diffusion. Facilitated diffusion is used to transport a variety of substances required for cellular survival, including glucose and amino acids. Osmosis is the movement of water across a semi-permeable membrane due to a water concentration difference. Osmosis follows the same principles as diffusion of any solute. For example, if two solutions A and B are separated by a membrane impermeable to the solute but permeable to water, and A contains a higher solute concentration than B, a driving force exists for water movement from B to A to equilibrate water concentration differences. Thus, water moves towards a solution with a higher osmolality. Osmolality is a measure of the total concentration of the discrete solute particles in solution and is measured in osmoles per kilogram of water, because it is much more practical to measure the volume than the weight of physiological solution the concentration of solute particles is typically expressed as osmolarity, which is defined as osmoles per liter. Osmolarity is equal to G into C, where G is the number of particles in the solution, that is osmos per mole, and C is the concentration, moles per liter. Osmolality is measure of total concentration of discrete solid particle in solution measured in osmoles per kg. Because it is more practical to measure the volume than the weight of the physiological solution, the concentration of the solid particle is expressed as osmolarity, which is defined as osmoles per liter. Osmolarity is equal to G into C. G is number of particles in solution, C is concentration. Consider the following example. What is the osmolarity of 0.1 mole per liter of sodium chloride? For sodium chloride, G is equal to 2. So, osmolarity is equal to 2 osmol per mole into 0.1 mole by equal to 0.2 osmol per liter, or 200 milliosmol per liter. Two solutions that have the same osmolarity are described as isosmotic. An isotonic solution is the one in which the volume of cells incubated in it does not change, implying that there is no movement of water in or out of the cell. Under normal condition, an isotonic solution is isosmotic with intracellular fluid, which is isosmotic with plasma, that is 290 milliosmol per liter. Not all isosmotic solutions are isotonic. A 290 millimolar solution of urea will be isosmotic 290 milliosmol per liter, but not isotonic, because urea is permeable to cell membrane and will diffuse inside the cell. This causes an increased concentration of urea inside the cell, which induces water influx and an increase in cell volume. Primary active transport is the transport of a substrate across the plasma membrane against its concentration gradient. It requires the input of cellular energy in the form of ATP. Proteins that mediate primary active transport are known as pumps, which use the energy derived from ATP hydrolysis to power the transport of substrates against their concentration gradient. The best studied example of primary active transport is the sodium-potassium ATPase sodium-potassium pump. The sodium-potassium ATPase generates low extracellular potassium and high intracellular sodium concentration. Another example of primary active transport is calcium ATPase, which clears calcium from the cytoplasm. Such calcium pumps are found on both plasma membrane and endoplasmic reticulum membrane. The short form ER stands for endoplasmic reticulum. Coupled transport or secondary active transport uses the energy of isotonic gradient, usually sodium, across the plasma membrane. Coupled transport still carries substrates against their concentration gradient, but transport is provided indirectly from the energy stored in the concentration gradient of an additional ion transported in the same cycle. For example, in a sodium coupled transporter system, sodium concentration is higher in the extracellular space than in cytoplasm. Therefore, sodium movement into the cytosol is energetically favored. Coupled transport mechanisms are divided into two groups. Co-transporters are also called symporter. They move solutes in the same direction. And then exchanges, also called antiporters, transport solute in opposite directions. Co-transporters and exchangers work only if both substrates are present. An example of a co-transporter is the sodium glucose transporter found in the renal proximal tubule and small intestine, which allows glucose absorption. An example of an exchanger is the sodium calcium exchanger found in many cells types and important in the regulation of cytoplasmic calcium. The exchanger transports 3 sodium in favor of 1 calcium, 3 sodium in for 1 calcium out, making it an electrogenic transporter. It is electrogenic because it makes a small contribution to the electrical potential across the membrane. Clinical correlation of this concept which I have told you, cardiac stimulants. The sodium pump is the target for a class of naturally occurring compounds from the wildflower digitalis purpurea or foxglove. These compounds have been used for almost 2 centuries as cardiac stimulant. These cardiac glycosides, including Oor and digitalis, inhibit the sodium-potassium ATPase pump. Oor is a poison found in a plant glycoside. Ion channels. Ions move quickly through protein pore in biologic membranes known as ion channels. Ions flow through these channels from one side of membrane to other down their electrochemical gradient. Channel proteins display 2 different conformational states, open or closed. The process that controls the transition between conformational states is called ion channel gating. Is the mechanism that controls the probability of a channel being in each of its conformational state. Voltage activated channels are opened and closed by the membrane potential. For example, a voltage gated sodium channel is closed at the resting membrane potential and is open only when the membrane potential is rapidly depolarized. Ligand activated channels are controlled primarily by binding of extracellular or intracellular ligands to the channel proteins. These channels are grouped into 3 categories: direct receptor channel complex, intracellular second messenger gated channel, direct G protein gated channel. In a direct receptor channel complex, the receptor for ligand is a direct part of the channel protein. The nicotinic acetylcholine receptor AChR is an example of this type of channel. In an intracellular second messenger gated channel, the binding of ligands to receptors activates a cascade of second messenger molecule, one of which binds to the channel protein in order to control channel gating. The cyclic guanosine monophosphate CGMP gated channel in a photoreceptor is an example. In a direct G protein gated channel, the binding of a ligand to its receptor activates a guanosine triphosphate GTP binding regulatory protein G protein that changes the conformation of the channel without involving second messenger system. For example, the cardiac inwardly directed potassium channel KACH, which allows the heart after vagus nerve stimulation, is gated by a G protein K-acetylcholine. Potassium acetylcholine channel is an example of G protein channel. Ion channels can select one kind of ion over another. Channels are often named according to the ions they prefer. Example: sodium channel, potassium channel, calcium channel. To account for the selectivity in certain voltage gate channel, there appears to be a narrow region in the channel pore that fits only on a particular ion. Ion channels provide a useful target for drug action. Lidocaine, an antiarrhythmic drug, blocks sodium channel in a use-dependent manner. Then higher the frequency of stimulation, external heart rate, the more that lidocaine blocks the channel. Ion channels are affected by diseases both directly and indirectly. Direct actions on the channel protein structure occurs as a result of genetic mutations of the channel gene. Indirect actions include abnormalities in the regulator mechanism required for channel function and in the development of autoimmune disease. Clinical correlation of what was said in the concept bridge so far on channels. Ion channel disease, cystic fibrosis, is an autosomal recessive disease that affects 1 in 2,500 individuals. It is an example of a direct effect on ion channel. The disease is caused by mutation in the cystic fibrosis transmembrane regulator gene, CFTR gene, which codes for the chloride channel gated by cyclic AMP. Chloride channel gated by cyclic AMP. In most cases of deletion of a single phenylalanine molecule, phenyl delta 508, prevents the channel protein from reaching the plasma membrane. In most cases, the deletion of the phenylalanine molecule prevents the channel protein from reaching the plasma membrane. This disease is caused by mutation in the CFTR gene, and this gene codes for the chloride channel gated by cyclic AMP. The drastic reduction in chloride channel results in thick mucus secretion that blocks airway, leading to death in 90% of the patients before they reach adulthood. Sorry for the error. It is the cyclic AMP gene. Myasthenia gravis is an indirect ion channel disease produced by autoimmune disease. Autoantibodies against the acetylcholine receptors lower the receptor concentration, causing lysis of the motor end plate. The decreased number of the nicotinic acetylcholine receptors results in smaller post-synaptic response and a tendency to block neuromuscular transmission. There is decreased number of nicotinic acetylcholine receptors in the post-synaptic area, and this blocks the neuromuscular transmission. Individuals with this disease experience weakness of skeletal muscle. So there is a autoantibody against the acetylcholine receptor, the nicotinic variety, in the postsynaptic area. Cell volume regulation depends on the total amount of intracellular solute. Following cell shrinkage, mechanisms that increase solute concentration are activated. This activation is achieved either by synthesis of smaller organic molecules, that is osmotically active molecule like sorbitol or taurine, or by transport of ions inside the cell through the sodium hydrogen exchanger or the sodium hydrogen chloride co-transporter. Increase the solute concentration inside the cell will induce water movement by osmosis and increasing cell volume. This happens during cell shrinkage. Alternatively, if the cell wall swells, transfer mechanism that extrude solute out or pump solute out of the cell, like potassium or chloride channel, potassium chloride co-transporter, will be activated. Because of the transfer mechanisms involved, cell volume regulation depends ultimately on the sodium potassium ionic gradient generated by the sodium potassium pump. Regulation of cellular pH at a constant level is critical for cell function. Changes in cellular pH can alter the conformation of proteins with the ionizable group, including a variety of enzymes and channels, thus affecting their function. Transport mechanisms that carry either hydrogen or bicarbonate are important for the maintenance of cellular pH. Transporters include the sodium hydrogen exchanger, which alkalinizes the cytosol, and potassium hydrogen exchanger in corneal epithelium, which acidifies the cytoplasm. Epithelia are sheets of specialized cells that link the body to the external environment. Epithelia are polarized at the structural, biochemical and functional level. This means that one side of the epithelial sheet contains different components and possess different properties from the other side. The side of the cell facing the lumen is called the apical side. The opposite side is the basolateral. The side of the cell facing the lumen is called the apical side. The opposite side is called the basolateral side. Transepithelial transport can be in the form of either secretion or absorption. Solutes can cross an epithelial cell layer by moving through the cells. Transcellular pathway or by moving between cells called paracellular pathway. Epithelia are classified as tight or leaky based on permeability of the paracellular pathway to ions. To understand how absorption through an epithelial cell layer occurs, consider the example of sodium chloride absorbing epithelium in the small intestine. The primary sodium entry pathway is on the apical side and varies with the tissue. It can be either a sodium channel or a transport such as sodium hydrogen exchanger or sodium coupled co-transporter like sodium glucose and sodium amino acid. Sodium channels on the apical membrane are membranes of amyloid sensitive sodium channel family. Sodium efflux across the basolateral membrane is performed by the sodium-potassium pump. Therefore, sodium enters the apical side and is secreted at the basolateral side, resulting in net transport of sodium across the epithelium. Chloride follows sodium movement across the epithelium through either the transcellular or the paracellular pathway, depending on the tissue. The transcellular pathway refers to ion movement through the cell layer, whereas paracellular pathway refers to ion movement between cells. Driving force for chloride movement through the paracellular pathway is the electrical potential generated by the net movement of sodium, that is, positive charge on the basolateral side. Alternatively, if chloride crosses the epithelium through the transcellular pathway, it usually enters at the apical side through the transporters like a chloride bicarbonate exchanger, sodium potassium 2 chloride co-transporter, and leaves the cell at the basolateral side through the chloride channel or the potassium chloride co-transporter. The activity of the sodium potassium ATPase on the basolateral side will result in the transfer of potassium ion inside the cell. Therefore, to maintain steady state ion concentration at the cytosol, the cell must have a mechanism to recycle the pumped potassium. This mechanism involves a variety of potassium channels located on the basolateral membrane. Secretions is conceptually more difficult than absorption, but the same principles discussed for absorption apply. The sodium potassium ATPase on the basolateral membrane pumps sodium out, potassium into the cell. Potassium is recycled back into the extracellular fluid through the action of potassium channel on the basolateral membrane. The sodium gradient generated by the sodium potassium ATPase is used to drive the sodium potassium 2 chloride or potassium chloride co-transporter on the basolateral membrane, resulting in net transport of chloride into the cell. The increased chloride concentration inside the cell causes chloride secretion through the chloride channel on the apical membrane, resulting in net chloride transport across the epithelial cell layer. The combined secretion of chloride into the lumen, that is apical side, and efflux of potassium through the potassium channel on the basolateral membrane results in a transepithelial potential that is more negative on the luminal side. This negative potential drives movement of sodium through the paracellular pathway towards the lumen. Intracellular calcium regulation plays a physiologically important signaling and regulator role in the various cellular processes. Cells have developed elaborate mechanisms to control calcium level and signals. Calcium signaling in the cytoplasm occurs through the rise in calcium level, which activate calcium binding protein that transduce the calcium signal into a cellular response. Therefore, maintenance of a low cytoplasmic calcium level is required for calcium signaling. A 20,000-fold concentration gradient exists for calcium across the plasma membrane. Furthermore, cells also contain intracellular calcium stores that are sequestrated in the endoplasmic reticulum, which contains high level of calcium. Calcium signaling occurs through a rise in cytoplasmic calcium level due to either calcium release from the endoplasmic reticulum or calcium influx from the extracellular space. Cells maintain low cytoplasmic calcium level by extruding calcium out of the cell using the plasma membrane calcium ATPase and sodium calcium exchanger, or by sequestrating calcium into the endoplasmic reticulum using the endoplasmic reticulum calcium ATPase. Cells increase their cytoplasmic calcium level in response to primary signals such as hormones or growth factors. Once the primary signal is received, calcium channels on the endoplasmic reticulum membrane or in the cytosol open, releasing calcium into the cytoplasm and transducing the primary signal into a cellular response. Channels on the endoplasmic reticulum membrane that mediate calcium release include the inositol 1,4,5-triphosphate IP3 receptor and the ryanodine receptor. Calcium influx from the extracellular space is mediated by different channel classes, including ligand-gated channels such as acetylcholine receptor and voltage-gated channels such as calcium channel in cardiac muscle. The clinical correlations: diseases associated with calcium regulatory defects like malignant hyperthermia, Brody disease, diarrheal disease, X-linked congenital stationary night blindness, and Lambert-Eaton myasthenic syndrome. A few we will discuss in this presentation. Malignant hyperthermia is a subclinical disease resulting from a genetic predisposition to react abnormally to volatile anesthetics such as halothane and muscle relaxants such as carbachol. Malignant hyperthermia is due to mutation in the ryanodine receptor leading to an overactive receptor. The mutated ryanodine receptor is especially sensitive to the aforementioned anesthetic, resulting in increased calcium release and sustained muscle contraction. Under severe conditions, extensive necrosis of muscle cells follows, leading to release of large amount of potassium, then cardiac arrhythmia and often lethal ventricular fibrillation. High calcium level will also lead to the continuous activation of the endoplasmic reticulum calcium channel ATPase and muscle contraction, resulting in increased heat production and hyperthermia. Vigorous exercise could also lead to abnormal muscle contraction in individuals with malignant hyperthermia. This condition can be treated with dandrolin, which inhibits the ryanodine receptor. Brody disease is an autosomal recessive mutation in the endoplasmic reticulum calcium ATPase channel, which leads to exercise-induced impairment of skeletal muscle relaxation. Diarrheal disease is a skin disease due to mutation in the endoplasmic reticulum calcium ATPase, leading to disruption of the cytoskeleton of the skin cells and loss of adhesion between these cells. X-linked congenital stationary night blindness is a recessive disease of the human retina due to mutation in the voltage-gated calcium channel, leading to defects in the glutathione release and neurotransmission, which impairs the function of the rod and cone cells in the retina. Eaton-Lambert myasthenic syndrome or Lambert-Eaton myasthenic syndrome (LEMS) is an autoimmune disease characterized by an increased number of LEMS antibody against the voltage-gated calcium channel, leading to defective neurotransmission and weakness of proximal muscles. So that was a session which I wanted to tell you about the cell biology and physiology. Hope that the session was useful to you, and the next session will be on cell signaling. And goodbye from ACPGMAT. Please visit the channel youtube.com/ACPGMAT. I am Dr. N.S. Mani from ACPGMAT, and for clarification and enquiry, please send your mail to acpgmat@gmail.com. And those who want to get the DVD by post, please send an email request with your correct postal address, and once the request is received, I will consider sending, depending on how many of you need the DVD of the past transmitted videos. The cost will be decided later depending on the number of the requests I receive, but it will be very minimum only. Since this is only a small venture, the cost will not be high. Thank you.